Right, so we're going to look at tissues and organs of the lymphatic system. This is what you're going to learn. The first thing you're going to look at are the components of the lymphatic system. When you talk of lymphatic system, what are we referring to? Which structures constitute the lymphatic system? We'll then look at the key functions of the lymphatic system, what the role. One of the things that are closely linked to the lymphatic system are the white blood cells. I will take this opportunity to take us through the different varieties of white blood cells, whether or not they are involved with the lymphatic system. I'll take a general approach to that so that we cover them at least holistically. From this perspective of anatomy, you'll still have a similar thing, I believe, in hematology or immunology or physiology, but from anatomy aspect, I'll also give you a highlight. We'll spend more time on looking at the structure and functions of the central and peripheral lymphoid organs. And in this lecture, we're going to understand what constitute the central and what constitute the peripheral lymphoid organs. Let's start with the first agenda there, components of the lymphatic system. And this first agenda will come to you in form of question, which I want you to respond to. You have a minute to respond to the question. You can scan the QR code on the screen and respond, or you can go to menti.com and enter the code there on the screen. All right, 1 minute is over. We can proceed. I can see what most of you have written. Almost everything, if not all, are actually correct responses to this particular question. That gives me an idea that you know what you're going to talk about in this particular session. When you talk of components of the lymphatic system, we're referring to many things. Top on the list, I want to start with white blood cells. We have particular white blood cells, which are part and parcel of the functions of the lymphatic system. And they serve key functions, as you're going to see. We also have lymphatic vessels. The lymphatic vessels carry lymph or lymphatic fluid around the body, from the periphery all the way back to the bloodstream, actually. So these lymphatic vessels are also found all over the body, and we'll be saying something about them. We also have what we call the diffuse lymphatic tissue. We are going to see what is this diffuse lymphatic tissue. We also have what we call lymphatic nodules. There's a combined term for number 3 and number 4. The combined term is mucosa-associated lymphatic tissue, MALT. And so we'll be talking about MALT. We do have lymph nodes, which most of you have written. We also have the spleen, and of course, the thymus and the bone marrow that most of you wrote. So these are the components of the lymphatic system. Second agenda is to look at the key functions of the lymphatic system. I will give you this again in form of equation. So the question is on screen. You can scan the QR code, or you can just enter the code and respond. All right, so we can close it. For those who are not able to access the code or enter Menti, I'm not so sure how to handle that. But we'll continue. Now, this is an important question, and let's spend some time here so that we can go forward. There are 4 key functions of the lymphatic system. One of them, top on the list, is on immunity, ability to prevent against infections. However, the way it operates to prevent against infections is very unique. If you think about it, immunity or defense against infection can be classified into different mechanisms. There's what we call the first line of defense. There's what we call the second line of defense. There's what we call the third line of defense. I'm not so sure whether you've been taught those ones in immunology, but we have those 3 lines of defenses. When you talk of the first line of defense and the second line of defense, we are talking largely about innate immunity. And in talk about the third line of defense, we are talking about adaptive immunity or acquired immunity. Innate immunity is the immunity you're born with. The first line of defense largely deals with barriers. We have anatomical barrier, we have physiological barriers, and we have biochemical barriers to infections. So that is first line of defense. Second line of defense deals with the inflammatory and cellular barriers. Those second line of defense are still part of innate immunity, although there are some components of this second line of defense which influence the third line of defense. Things like complement system, for example. When you talk of cellular, or rather cellular barriers and inflammatory barriers, when we talk about cellular barriers, we're largely talking about the act of phagocytosis, cells, blood cells eating or engulfing the pathogens. And when we talk about inflammatory barriers, we talk about inflammation in particular, which can be acute inflammation, chronic inflammation, inflammation mediated by complement system and the like. So that's generally the breakdown of first line and second line of defense. Now, first line of defense, I've told you, is either anatomical barriers to infections, physiological barriers to infections, or biochemical barriers to infections. When we talk about anatomical barriers to infections, we're talking about things like skin or the mucosal membrane, mucous membrane, and the like. They have to prevent organisms from penetrating that constitute anatomical barrier to infections. That cannot be a function of the lymphatic system. Yes, lymphatic system will have the white blood cells, but they engage in mechanisms which are not necessarily anatomical barriers. They could be cellular barriers, or they could be largely part of the adaptive immunity, but not anatomical barriers. So the 9 of you are the ones who have gotten that question right. Anatomical barriers to infections cannot be a role of the lymphatic system. Site of hemopoiesis is actually a role of the lymphatic system. We know that hemopoiesis is formation of blood, and we know that blood is formed within the bone marrow. So bone marrow is actually very key, a key component of the lymphatic system, but also, yes, site of production of white blood cells, actually all blood cells. But other than bone marrow, the spleen and the lymph nodes are also important in hemopoiesis. The spleen will participate in formation of blood before the child is born, so before birth. After birth, the spleen can participate, but if the bone marrow fails, and you'll be seeing cases of this kind of condition. In diseases of the bone marrow, the spleen and lymph nodes can take up the role of hemopoiesis, and we call that extramedullary hemopoiesis, to distinguish it from intramedullary hemopoiesis, which is basically hemopoiesis within the bone marrow. So yes, organs of the lymphatic system can participate in formation of blood. Majority of you picked C as an exception, but these are very key function of the lymphatic system as well. I know you've not done GIT physiology, but when nutrients are being absorbed from the intestines into the circulation, carbohydrates and proteins, vitamins, water will be absorbed into the bloodstream from the intestines. However, fat and fat-soluble vitamins do not go to the bloodstream. They go to the lymphatic channels, which we call lacteals. And then these lymphatic channels redirect this fat all the way to the junction between the left subclavian vein and the left intermedullary vein, somewhere up in the neck there, the left side. The lymphatic channel that does that is called the thoracic duct, which we discussed, I think, last week. So eventually, fat from the digestive system will go through the lymphatic channels to go to the bloodstream. The main purpose of this approach is so that it can evade overloading the liver with a lot of fat. Blood from the intestines usually go to the liver, as we saw even last week when we discussed fetal circulation and its changes at birth, blood from the intestines may go to the liver. You don't want this blood going to the liver to deliver a lot of fat to the liver tissue because it's likely to cause some injury. Therefore, fat is deviated away from the liver, goes to the bloodstream. So when it enters the venous circulation, it will go to the heart. Then the heart will pump it to all parts of the body, of course, including the liver. But in so doing, we'll have reduced the concentration of fat that is loaded to the liver at once. Tissue fluid balance is also a key function of the lymphatic system because when blood extravasates from the capillaries into the tissues, for that blood, the fluid that has extravasated the capillaries may go back into the blood vessels or may enter the lymphatic channels. So it's a mechanism of ensuring that we don't have a lot of fluid within the tissues. And therefore, this is actually a way of controlling tissue fluid. Excess accumulation of tissue fluid will give us what we call edema, and that's what we're preventing from happening. So those are the key functions of the lymphatic system. Now we can talk about the white blood cells. Somebody's asking, what is the second line of defense? So I said the first line of defense are barriers, anatomical, physiological, biochemical barriers. Second line of defense would now apply to what we call cellular and inflammatory barriers. Then third line of defense would be the adaptive immunity. And you know that in adaptive immunity, you may talk about humoral immune response, or you may talk about cellular immune response. I believe you've handled that in immunology, and if not, you'll be handling them soon. Let's look at the white blood cells. We'll also spend some time here. Now, understand that blood is a tissue, and blood has three major components, or let me say two major components. We have the plasma, which is the fluid component of blood, and we have the cellular elements of blood, or what we call the formed elements of blood. The cellular or the formed elements of blood can now be grouped into three. We have the red blood cells, which are the majority, occupying about 42 to 45% of the whole blood, generally speaking. And in other quarters, that is what is called the hematocrit, or the packed cell volume. Then we have what we call the platelets, which are responsible for clotting. The third entity of the formed elements are the white blood cells, which are important in immune functions. I want to ask to now outline these white blood cells. So even though they'll constitute less than 1% of whole blood, basically speaking, because if you look at whole blood, plasma will occupy about 55% of the whole blood. The red blood cells occupying about 45% of the whole blood. So you remain with hardly any percentage, that hardly any percentage, or less than 1%. In other quarters, this less than 1% is called the Buffy coat, B-U-F-F-Y coat, C-O-A-T. The Buffy coat is where the white blood cells and platelets belong. Albeit the fact that they're very contributing almost less than 1%, they are very important. And let me just now give you a breakdown of the white blood cells. We divide white blood cells into two categories. We have what we call granulocytes, and we have a category we call the agranulocytes. This distinction is based on the presence of granules within their cytoplasm. So the ones which have granules, these granules contain chemicals which are used in defense. It could be histamine, leukotriene, and the like, several types of chemicals which are present within these vesicles, which we are calling granules. So the cells which have a lot of granules in their cytoplasm will be called granulocytes. And the ones which do not have a lot of granules in their cytoplasm are now called the agranulocytes. Let's begin with the granulocytes. Three categories of granulocytes. We have what we call the neutrophils. The neutrophils are the majority of the white blood cells. About 50 to 70% of all white blood cells are actually neutrophils. There are some authorities that will give you different percentages. It's okay and acceptable. So sometimes you may see 40 to 65 or 45 to 75. Key thing here is that neutrophils constitute majority of all the white blood cells. When you start your clinicals, you'll be calling this thing the differential count. The percentage represented, or rather percentage of all the white blood cells that is occupied by a single white blood cell is what we call the differential count. The differential count is important in telling us which type of white blood cell has increased in number. And that helps us to make some clinical diagnosis. It could be in line of cancers, or it could be in line of infections, but we are able to tell. That's why when you have fever, for example, and you go to hospital, they'll request for what we call full hemogram. Full hemogram will have the HB there, so maybe it's 12, that's the hemoglobin level. It will also have the white blood cell count. You will have the absolute count as well as the differential count. The absolute count is good, but doesn't tell us much. And that is why we look for the differential count. So you might see that, yes, you have a lot of white blood cells, but is it all the white blood cells affected? Maybe no. That's why we need to know which white blood cell is increased. And that's why we want to know the differential count. So it's important for you to capture the differential counts of the white blood cells. Neutrophils are the majority. Their mechanism of action, mechanism of defense, the key mechanism of defense is by phagocytosis. So these are phagocytic cells. They are very key as part of what we call the second line of defense against infections. They are the ones which are responsible for providing immunity against extracellular infections and especially extracellular bacteria. So if you go to hospital and the levels of neutrophils are very high, most likely you have a bacterial infection, most likely. Not all the time, most likely. The second category of the granulocytes are the ones we call the eosinophils. Eosinophils are not that much, about 1 to 4% of all white blood cells are actually eosinophils. The mechanism of action of eosinophils is to release their granules. I told you that granulocytes have their granules. They release their granules. And through that, they offer immunity against helminthic infections, these large infections like worms. They may also offer immunity against protozoal infections by releasing their granules to these particular organisms. Basophils are also key. Now, basophils are probably the least of all the white blood cells. They are the fewest or the least of all the white blood cells. Their mechanism of action is by degranulation again. And their granules are associated with allergic reactions. So the basophils are the ones which mediate allergic reactions. If you are asthmatic, you may want to blame the basophils to be specific. We call them basophils when they're in the bloodstream. But when they leave the bloodstream and enter tissues and probably dwell there, they change their name from basophils to mast cells. But the key thing here is that they act by degranulation, releasing their granules into the extracellular space. Most of the time, this will be acting through paracrine fashion, affecting the cells within the neighborhood. So for those who are asthmatic, it's more of the mast cells of the respiratory tree, which are hyperactive and therefore making the bronchus hyperresponsive. Great. So these are the granulocytes. How about the agranulocytes, the cells that do not have granules within their cytoplasm, or at least the granules are not that main? We have what you call the monocytes, called so because they have a single nucleus, and this is to distinguish them from this type of white blood cells, which have nuclei with multiple morphology. So you can call them the polymorphonucleans, white blood cells. Their nuclei have multiple morphology. Some are binucleated, some have bilobed nucleus, trilobed nucleus, quadrilobed nucleus. So the nuclei have multiple morphology. The term we use is polymorphonuclear white blood cells. Applies to this one. And so to distinguish them from those, these ones have a single clear outline of nucleus. We call them monocytes. They constitute about 2 to 8% of all the white blood cells in the body. And generally, their key mechanism of action is also by phagocytosis. Important to note is that these are the cells that constitute the macrophages. So we call them monocytes when they are in the bloodstream. When they leave bloodstream and go into tissues, they're the ones that constitute the macrophages. And you remember from anatomy 1, I think, that we have different types of macrophages in the body, depending on the tissue of concern. So you may call them microglial cells if you're in the central nervous system. You may want to call them monocytes in the bloodstream. You may want to call them Langerhans cells in the skin. You may want to call them osteoclasts in bone tissue, pneumocyte type 3 when in the lung and the like. I believe you remember the list of the macrophages according to their tissues of residence. Important to note is that macrophages are antigen processing and presenting cells. Their key role is to break down, or rather to engulf the antigen. Let's consider a bacteria here to be the antigen. Engulf that bacteria, break it down, split into smaller pieces, then present those smaller pieces to the lymphocytes. So we consider macrophages are antigen processing and presenting cells. How about the lymphocytes? Lymphocytes are of 3 different types, but in total, they constitute about 20 to 40% of all the white blood cells. So that's their differential count. There are 3 types of lymphocytes. We have the B lymphocytes, or the B cells. The B lymphocytes are called so because they mature within the bone marrow. Now, the B here, in terms of entomology, does not quite refer to bone marrow itself. That's coincidental, but a good coincidence. The discovery of the B cells was after a study that was done in birds, aves, and birds have a region within them, which we call the bursa of Fabricius. So the B there was for the bursa of Fabricius. In humans, we don't have the bursa of Fabricius, but the equivalent of this is the bone marrow. So it's still BB applies. B cells are formed in the bone marrow, but they also mature in the bone marrow. Their training takes place within the bone marrow. The mechanism of action of B cells is that when they are stimulated, you may have different subtypes of B cells. That those that we call the B memory cells, which keep immunological memory of an infection. Then there are those that we call the plasma cells. The plasma cells act by producing antibodies. And we have different types of antibodies. Antibody G, antibody A, antibody M, antibody E, and antibody D. Those are different types of antibodies, or what we call immunoglobulins. Now, because B cells offer defense by production of antibodies, immunity by antibody production is what we call humoral immunity. And this is an important part of adaptive immunity. It's a very important part of acquired immunity, humoral immunity. So the B cells are the center of humoral immune responses. So the plasma cells are actually the cells that will eventually produce it's a subtype of activated B cells that now actually produces the antibodies. Then you have the T cells. T cells are called so because they mature within the thymus. Take note that they will all be produced within the bone marrow, but then these ones mature within the thymus. We call them the T cells. So all blood cells are produced in the bone marrow, credit to the bone marrow, but the T cells do not mature there. Their training does not occur in the bone marrow. Their training occurs in the thymus. And so T for thymus. We have different types of T cells, different varieties, and I think that will be covered better in immunology, not in this anatomy class. Just to give you examples, you may have what you call the T cytotoxic cells, the T memory cells, the T regulatory cells, the T helper cells, among others perhaps. So those are different subtypes of T cells according to their specific functions. In particular, the T cytotoxic cells are important because they offer immunity by killing other cells. They offer immunity by killing other cells. And that's why we call them the T cytotoxic cells. These T cytotoxic cells, therefore, are at the center of what we call cell-mediated immunity or cellular immunity. And because of that, therefore, we say that the T cells are actually at the center of immune response, which you're calling cellular immunity. And basically, those are the two arms of adaptive immunity. You either have humoral immunity or cell-mediated immunity. They're not the only cellular players of cellular immunity, but that they're really at the center of it all. This is immune response by killing infected cells. This is perhaps one of the key mechanisms of immune response against intracellular infections. So infections which are inside the cell, for example, viral infections, or some bacteria which are intracellular. This is the immunity that will help them. While humoral immunity will be more for extracellular infections, the bloodstream. All right, the third category of lymphocytes, what we call the natural killer cells. They are known to kill cells indiscriminately. That's why we call them the natural killer cells. Great. So this is the breakdown or a general highlight of the white blood cells. I have two questions for you here, and I'll want you to unmute your mic and answer if you know the answer. First question, if you do a full hemogram, basically somebody is sick and they're going to hospital and they've requested a full hemogram. And in the full hemogram, the result shows that the lymphocytes are 90% of all the white blood cells. What does that tell you? Anyone? So feel free to unmute your mic and respond.
It means the individual has an infection.
Okay. Do you want to elaborate on what kind of infection could that be?
Bacterial infection.
Okay. Why do you think that would be bacterial infection? Paris, why should that be bacterial infection?
Because the infection, it's in the blood.
Okay. Any other person with a different opinion?
Could it be cancer?
Okay. Why do you think it could be cancer?
Because of the cytotoxic cells, T cytotoxic cells, and natural killer cells that try to kill the cells by themselves because the cells are overgrowing.
Okay. Any other person with a different opinion? Yes, Jerisha?
I think it should be viral infection.
Okay. Why do you think it should be viral infection?
Because trigger the lymphocytes and the bacterial infections trigger the neutrophil.
Okay, fair enough. So thank you for the responses. The truth of the matter is that it could be anything of those things you mentioned, but there should be some guideline into what makes you think about one direction and not the other. Let's start with the possibility of it being a bacterial infection. If you get a full hemogram that is 90% lymphocytes, it is very unlikely to be a bacterial infection. Could still be possible, but very unlikely. Because we know that bacterial infections will be mainly extracellular infections, and therefore would expect neutrophils to be the key players in that particular field. Unless it's a long-term infection, these chronic long-term infections where they tend to be intracellular, like mycobacteria, may not give you neutrophilia, but may give you what we call lymphocytosis. Lymphocytosis is the term we use for increased lymphocyte count. Neutrophilia is what you use for the increased neutrophil count. So you can say neutrophilia, eosinophilia, basophilia, and the like. But this other side, we don't talk about lymphocytopenia, we talk about lymphocytosis. So if it is bacterial infection, we would expect more of the neutrophils increasing, and especially in the acute setup, rather than the lymphocytes. A viral infection is most likely for lymphocyte count that increases. However, 90% is quite high for a response to lymphocyte, to viral infection. We would expect it to be maybe 50%, 60% there. 90% is quite high. Not impossible, can still be a viral infection. Just the same way it can still be a bacterial infection that has been long-standing, indolent bacterial infection. But again, very unlikely. The chances are the scenario I've given is actually a cancer scenario. But the explanation Boniface gave is not very accurate for why he thinks it should be cancer scenario. So in response to cancer, yes, cytotoxic cells are the ones which respond to by killing the cancer cells. But that does not quite mean that they usually increase their number quite a lot to that significant level. Now, the reason why this could be cancer is because the blood cells themselves could be the ones which are cancerous. And that's the point here. It's not that they have increased in number to fight the cancer cells. No, but it's themselves affected. A lymphocyte count that is over 90% of white blood cell count is most likely a leukemia picture. And that's basically cancer. It's most likely a leukemia picture. And we have different types of leukemias. You may have leukemias which are predominantly of the lymphocytes. So we call them lymphocytic leukemias. Or leukemia which are largely of this type, the granulocytes and the monocytes, and perhaps the red blood cells. And those are the ones we call the myeloid leukemias. When you discuss bone marrow, you'll understand the difference between myeloid and lymphoid series of cells. All right. So let me just say something about the lymphocytes. Lymphocytes are the definitive cell types of the lymphatic tissue and the lymphatic system. All the other white blood cells are considered supporting cells of lymphatic system. But lymphocytes are the primary cell types of the lymphatic tissue. I've already told you that you have the B cells, T cells, and the natural killer cells. Their variety may not be distinguishable from full hemogram, so we usually use other laboratory ways of distinguishing them. And one of the ways of distinguishing them is by looking at the type of antigens they express on their cell surface. This type of antigens expressed by white blood cells are called cluster of differentiation, commonly abbreviated as CD. Let not that confuse you with another CD that you might be knowing. So cluster of differentiation antigen refer to specific types of antigens that particular subtypes of white blood cells display. I have put this slide here, not for the sake of you cramming. I also don't know many of them, but for the sake of you understanding the concept that we may have different types of white blood cells. These white blood cells express particular types of antigens on their surface. We can use the type of antigens expressed on the surface of white blood cells to know them. And so commonly there's some particular antigens that will be unique for particular cells. For example, when you look at this list, you'll see that CD4 is almost unique to monocytes and the T helper cells. And so occasionally we'll send patients for CD4 count. We know that we are targeting, we want to know how much of their lymphocytes are actually T helper cells, or how much of the white blood cells we have monocytes then. So there's some particular molecules which will be unique to particular cells. There's some particular molecules that cut across the board. For example, you can see CD45. Almost all white blood cells have CD45. So when you see that CD45, we know this is a white blood cell. Some of them will not have, and especially the stem cells for white blood cells, will not have the CD45. So when we see a white blood cell that does not have a CD45, again, there's a place we put it. We know that this is this subtype of white blood cells. You'll be told more about the process of formation, or the term has disappeared, but the process of proliferation and maturation of white blood cells. And so you'll know which molecule is at which point in time on the maturation ladder for white blood cells. But this is an important slide in terms of making you understand why we order some particular tests for some particular disorders or particular diseases. We want to know some markers. For example, if this was cancer, then I'll run what we call flow cytometry so that I know which cluster of differentiation is being expressed by these tumor cells. So if I see a particular cluster of differentiation, I'm able to know the cell type affected, the cell type that's abnormal in this particular cancer type. All right, so those are white blood cells, but let's talk about lymphatic vessels. So what are lymphatic vessels? I told you that when blood or fluid leave the capillaries to go to the tissues, the fluid may go back to the capillaries at the venule end, the venous end of the capillaries, and then enter into veins. But not all fluid will do that. Some fluid that has extravasated, instead of joining the capillaries to go to the veins, they'll join the lymphatic vessels. So lymphatic vessels will begin as network of capillaries within loose connective tissue generally. We have a lot of them in the skin and perhaps mucous membranes, like the membrane of the digestive system, the membrane of the respiratory system, and the like. There are several in the body, but they're basically the ones which carry lymphatic fluid. Now, usually at some junctions, the lymphatic vessels enter lymph nodes. They just pass through lymph node, and so you have a vessel entering a lymph node and another vessel arising from the lymph node to go to somewhere else. The vessel that enters the lymph node is called the afferent lymphatic vessel to that lymph node, and the one exiting is called the efferent lymphatic vessel. Of course, when they pass through a lymph node, it means that we want the lymph node to filter what is within this particular lymphatic vessel. So for example, here, let's consider this to be the systemic circulation, where the lymphatic vessels have now picked lymph from the tissues, goes back to at some point their lymph node intervening. So the afferent vessel will enter and the efferent vessel will leave. This efferent vessel may also be afferent to another lymph node. So you may have multiple chains of lymph nodes along the path of a lymphatic vessel, but eventually the lymphatic fluid will go back to the circulation through the thoracic duct. Important to note is that lymphatic vessels form channels through which interstitial fluid can go back to the circulation, also through which the white blood cells which exited the blood vessels can also go back to the bloodstream. Of course, we've also mentioned that it's also a channel, important channel for fat absorption in the intestines. So let's now look at the last part of this lecture, where we're going to talk about the structure and functions of the central and peripheral lymphoid organs. Maybe we don't know what is central and what's peripheral lymphoid organs. So let's start there. These organs, I've already given you a list of them. Which of them are we considering as central and which one are we considering as peripheral, and why? Central lymphoid organs are also known as the primary lymphoid organs. They constitute the site of production, training, and maturation of the lymphocytes. So it's a site of production, it's a site of schooling, the site of maturation of the lymphocytes. Generally speaking, the central lymphoid organs will have either the B cells or the T cells. Usually, there'll be territories where it's just a B cell territory or territory where there's a T cell territory. When you talk of central lymphoid organs, we are referring to bone marrow, which will be housing the training and maturation of B lymphocytes, and thymus, which is the site of training and maturation of T lymphocytes. Those are the only central lymphoid organs. How about the peripheral lymph organ? Everything else will be peripheral lymphoid organs. So peripheral lymphoid organs are also called secondary lymphoid organs. These are the sites of interaction between the immune cells and the pathogen. In simple terms, they are the battlefield, the point where fighting takes place between the immune cells and the infectious agents. There are also important sites of clonal expansion. What is clonal expansion? It simply means multiplication of one type of a cell. So for example, if there was a T cell that went to thymus and it got exposed to, let's say, this particular viral infection, and so it developed molecular mechanisms of identifying these infections or cells with this infection. If it's only one, it has already been trained to recognize that particular infection. Let's say it's been trained to recognize cells which are infected with the TB. The next time you get TB, this cell will be sensitized. It will be sensitized to proliferate after its own kind. It will give rise to cells of similar kind, cells which are able to recognize TB. Now, that proliferation of a cell type that has already committed to a particular role is what we're calling clonal expansion. It simply means numerical increase of a cell of one particular type, one clone, one type. So peripheral lymph organs are sites of clonal expansion. Remember that. They will contain both B cells and T cells in one particular organ. So there are different types of peripheral lymphoid organs. We have the mucosa-associated lymphatic tissues, tonsils, lymph nodes, and spleen. I want us to go through them. Let's begin with the diffuse lymphatic tissues. So this is an image obtained from the wall of the intestines, most likely the large intestines. The surface will be this side, so the lumen will be lower left there. So this is basically the mucosa with the epithelium. It's highly folded with some glands. Those glands, we'll discuss them later. But what I want you to notice that this region where we see hypercellularity, and those cells are quite close to one another. Those are basically lymphocytes. It's a cluster of lymphocytes. This cluster of lymphocytes, we cannot even tell how far it extends. If you look at this region, we can still see some cells. We can't tell how far it extends. Yes, you have a big cluster here, but we still have extensions on the other side. So this is what we call the diffuse lymphatic tissue. A diffuse lymphatic tissue is an amorphous accumulation of lymphocytes within loose connective tissues. They do not have a definite boundary, neither do they have a capsule that surrounds them. So they're just present all over. We find this kind of lymphatic tissues within the lamina propria of digestive system, respiratory system, and genitourinary tracts. Lamina propria is the loose connective tissue beneath the epithelial lining of these regions. They constitute part of what we call the mucosa. We'll be discussing that more, perhaps in 2 weeks' time. Strategically, they are located to intercept antigens and initiate immune response because they are located within mucosal surfaces. Now, they may go with other unique names. For example, we call them the mucosa-associated lymphatic tissues, MALTs, because they are found within the mucosa. So we call them the mucosa-associated lymphatic tissue. And depending on the organ of location, it may have another name. For example, the MALT located within the digestive tract is sometimes called GALT. And GALT here will stand for gut-associated lymphatic tissue. The ones found in the respiratory tree may be called BALT. And BALT here stands for bronchus-associated lymphatic tissue. So they're part of MALT. Other than the diffuse lymphatic tissues, we also have what we call lymphatic nodules. What are lymphatic nodules? Look at this. So this is, again, epithelium. This is of the esophagus. So this is epithelium of the esophagus. Stratified squamous non-keratinizing epithelium. Deep to it is the lamina propria. Then we see this accumulation of lymphocytes. So we agree, yes, we do not have a covering capsule, just like the previous one. But there's something unique about this that we didn't see in the other one. This one has a discrete boundary. We can tell that actually this accumulation is reaching somewhere there. We can actually surround the boundary vaguely, but at least we can say that there's a boundary. Unlike the other one where there was no sharp boundary. So lymphatic nodules are discrete. They have a sharply delineated boundary. We can see the boundary, but the boundary is not a capsule. Usually, they are contained within a meshwork of some supporting cells. There are some supporting cells found within this that we call the reticular cells, which hold the lymphocytes together. You might be wondering what makes them be together like that. We have a meshwork of reticular cells that hold them in that particular area. Lymphatic nodules also form part of the MALT as well. So when you talk about mucosa-associated lymphatic tissue, it could be the diffuse lymphatic tissues or the lymphatic nodules. There are some specific aggregations of lymphatic nodules that may have other names. For example, we have aggregation of lymphatic nodules around the pharynx. And that's what we call the Waldeus ring. We talked about that some time back. The ones found on the tongue will be called lingual tonsils. The ones found on the soft palate is called the palatine tonsils. The ones found on the tubal ridge is called the tubal tonsils. And the ones found on the roof of the nasopharynx is called the pharyngeal tonsils. When they are swollen, we call them the adenoids. So basically, those are lymphatic nodules, which are aggregated together. You have multiple aggregations of lymphatic. So you have something like this and another one here and another one here. They are in aggregations. In the small intestines, we have several lymphatic nodules, which are again aggregations, and they constitute part of what we call the Peyer's patches. So Peyer's patches are lymphatic nodules present specifically in the ileum. They are more in the ileum. The appendix also has several lymphatic nodules within it. It's actually a lymphoid organ, we call it. Now, when we raise the bar of the lymphatic nodule, so I'll not discuss tonsils because I've told you what tonsils are. They're just aggregations of lymphatic nodules without covering capsule. We call them tonsils. How about lymph nodes? Lymph nodes are small bean-shaped encapsulated structures that are interposed along the path of lymphatic vessels. You've noticed that we've not talked about lymphatic vessels for the diffuse lymphatic tissue and the lymphatic nodules because they don't intercept vessels. If anything, they may only have efferent lymphatics, but they don't have afferent lymphatics. However, the lymph nodes do have afferent and efferent lymphatic vessels because they intercept the lymph vessels. Very important. So their role will be to filter the lymph that percolates through them as that lymph goes to the bloodstream. Now, we have different aggregations of lymph nodes. We may have lymph nodes all over the body, but there's some regions with several aggregations. For example, in the axillary region, we have several lymph nodes to the level that we call them axillary group of lymph nodes. In the intestines, we have several of them. We call them mesenteric lymph nodes. In the groin, we have quite a number. We call them inguinal lymph nodes. So lymph nodes exist in groupings, and they constitute a group of lymph nodes. Of course, in the cervical group of lymph nodes, we also have many. If you remember some point when you're young that you occasionally have something swelling in your groin, it will be linked to perhaps an injury you have on your big toe or something like that. Those are actually lymph nodes which are swelling because of having intercepted some infections from the breach that is on your feet. Moving on, we have the spleen. The spleen is the largest peripheral lymphoid organ. It is located in the abdomen, usually on the left side, what we call the left hypochondrium of the abdomen. The left hypochondrium is this region below the left hemidiaphragm. We'll discuss more about abdominal regions when we discuss the abdomen. It lies usually behind the stomach. It also lies in front of the posterior dome of the left hemidiaphragm. And I'm saying that so that we understand that the diaphragm goes much lower than the level of the spleen on the posterior dome. And that means that sometimes you may have injuries, penetrative injuries that injures the spleen, but also injures the pleura because at the same horizontal level, they all exist. Grossly, the spleen has a smooth surface, which is usually posterior surface related to the smooth dome of the diaphragm. So we call the diaphragmatic surface, but also has a rugged visceral surface, which is related to abdominal organs. And there are 3 or 4 key abdominal organs related to the spleen. So there's region for the stomach. There's a region occupied that is related to the left kidney. And there's a region that is related to the colon. So usually it is the junction between the transverse and the descending colon. We call it the left colic flexure. So this is a region for the left colic flexure. This is a region for the left kidney. This is a region for the stomach, more so the fundus of the stomach. And somewhere around here will be the tail of the pancreas, making that particular impression, usually extending from up there, also up to somewhere there, the pancreatic tail. This region here is called the hilum of the spleen. The hilum of the spleen is the one that receives the splenic artery, splenic vein. Important to note is that spleen is the only lymphoid organ involved in filtration of blood. Lymph nodes will filter lymphatic vessels, or rather lymph within lymphatic vessels. But spleen filters blood. So it's very important. It's the only one. And because of that, it's very important in offering defense against blood-borne infections. The spleen has a covering capsule, just like the lymph node. They have covering capsule. And the capsule of the spleen is very thick and sends in some trabecula that enter into the spleen. This trabecula carry within some blood vessels. But within the parenchyma of the spleen, we have 2 types of histological zones, what you call the red pulp and the white pulp. We will talk more about the spleen histologically when we look at it practically on a microscope so that we know what is red pulp and what is white pulp. But in a nutshell, the white pulp are representative of the lymphatic nodules. So it's like a large encapsulated organ with multiple lymphatic nodules within it. So the white pulp represents regions occupied by the lymphocytes, and they're like the lymphatic nodules. The red pulp refer to regions of the spleen that has every other cell of the bloodstream. So red pulp contains everything that is usually in the bloodstream, while white pulp contain the lymphocytes in form of nodules. We look at it histologically. But what are the key functions of the spleen? The spleen is viewed to have both hematologic as well as immune functions. In terms of hematologic functions, the spleen is important in filtration of blood. It acts as a filter by removing old red blood cells. We know, or maybe we don't know, that red blood cells stay for about 90 to 120 days, then they are destroyed and removed from the body. So the spleen is important in doing that. Other than that, the spleen is also important as a reservoir for red blood cells. So usually it is able to hold a lot of red blood cells within it and may be releasing them into the circulation when need be. The spleen is also an important site of hematopoiesis. I already mentioned that before birth, the spleen is important in forming blood, and even after birth, the spleen participates in extramedullary hemopoiesis. Storage and release of platelets is an important function of the spleen. It's also able to hold a lot of platelets within it and release them to circulation. And that's why when a patient has the spleen removed, there'll be a lot of platelets in the circulation rather than now being held up. And that may cause some problems. These people will tend to have clotting tendencies because they have a lot of platelets now in the circulation. How about the immune function of the spleen? The spleen has several immune functions, but basically, remember, it's a site of initiation of immune responses to some antigens, especially blood-borne antigens. So this is the primary site where these responses are initiated. It's a key site of antibody production by the B cells. So once this immune response has been initiated, the B cells respond by production of antibodies. Spleen is also an important site of removing antibody-coated bacteria. So the bacteria that have been coated by antibodies following humoral immune response are also removed, destroyed and removed by the spleen. It also contains the other white blood cells, which perform immune surveillance. They just watchmen, they check, are we okay? Is there something wrong? And perhaps if you also have cancer cells, they remove those cancer cells. That's what immune surveillance is all about, identifying and eliminating potential threats in the system. So these are the key functions of the spleen. We will talk more about the spleen when you look at it histologically. We look at the structure. That brings me to a question and an assignment that I want to give you as we close. So the instruction is this: for the central lymphoid organs, bone marrow and thymus, of course, I want you to do a write-up. You're going to do this write-up in a group. So it's a group assignment. Not more than 300 words. You'll do a write-up on the structure, functions, and disorders of the central lymphoid organ. You will select one person to send that write-up to me by email, and you CC the rest of the members of the group before our next class. Our next class is on Friday next week. So you send before Friday next week. That means you have up to Thursday at 23:59 hours. All right. Thank you very much. We will close there, but I'll take questions if you do have.